Hi, everybody. This is Aditya Nain, and I'm in conversation today with Karan Shah, Dr. Karan Shah. Karan's a bone biologist and cancer researcher at Sheffield University, and he's going to talk to us about a bunch of different things, but specifically about his work in cancer and cancer research. So, Karan, maybe just talk a little bit about yourself, and we take it from there.
Yeah, so as Aditya said, I work at the University of Sheffield as a postdoc, and my work focuses on understanding how cancer interacts with bone in particular. And I've been here since the last 10, 11 years now, working as a bone biologist to start off with, and then moving into cancer. I came here to do my master's in 2008, and since then I did my PhD and a couple of postdocs here. So yeah, I've been in Sheffield for a while.
Okay, awesome. So, I want me and other people to try and understand what cancer research entails and the kind of work that you do, so that it's sort of made understandable for people who don't know much about it. Okay, so why don't you tell us what's on the slide?
Yeah, so my research focuses breast cancer in particular, and this graph here just highlights that when breast cancer spreads to different organs, bone is makes up about 50% of the chunk of it. So normally breast cancer spreads to bone, lungs, brain, and liver, and sometimes other places. But bone is the major organ where it goes to. And when it does spread to bone, it becomes incurable. Actually, when it spreads to anywhere, it's actually incurable. And therefore, we are focusing on understanding the mechanisms by which breast cancer actually interacts with bone and try to understand if we can stop it or delay it to improve the outcome for patients.
Okay, all right. So before we get into even that, I mean, I'm sure that's extremely simple for you, and it lays the foundation. But before we get into even that level of simplicity, could you just tell us a little bit about what cancer is? So for a complete layperson, how should they understand cancer as being different from, let's say, any other disease?
Yes, so cancer is basically the uncontrolled growth of cells. So all our cells in the body undergo multiplication, and after a particular time, the signaling suggests that they should die or undergo apoptosis. So when they stop dying, that's when they grow out of control. That's what we call cancer. And the problem with this is that it messes up a lot of different systems in your body when it does that, and therefore it is as deadly as it is.
Okay, so do you mean any cell can be cancerous?
In theory, yes.
Okay,
Any cell can convert to being a cancer cell.
and that happens when
So
it
there could
refuses
be a
to
lot of
die.
stimuli which can promote your chances of making a cell into a cancer cell, such as smoking, any kind of external damaging stimuli,
Hmm.
smoking, sunlight, radiation, drinking. All these environmental factors can kind of damage your cells, and when they damage your cells, they might mutate the genetic material of your cells, which
Hmm.
might make them lose the control of growth, and
Hmm.
that's therefore when they start multiplying uncontrollably, causing
Oh,
cancer.
so okay, so if I have to sort of convert that into the language that I usually think in, so it's there's nothing sort of qualitative about the cells that makes it cancerous, but it's more a question of when it multiplies quantitatively and in a manner that cannot be controlled anymore. Is that what it is?
Not completely, because there has to be a qualitative change to make it multiply quantitatively in the first place.
Right.
And that
Okay.
quantitative change is via the external stimuli, or maybe some inherent factors that you might have as well,
Mm.
genetic predisposition.
Okay, so it can be genetic predispositions or something external, which causes some sort of qualitative difference within the cell, which leads to a quantitative difference, which is the uncontrolled growth. So basically, cancer is cells that refuse to die.
That's true, yeah.
Okay, and why is that an issue?
Well, because normally in your body, you have a set function for an organ. So
Hmm.
if you, for instance, if you have, I don't know, if you have lung,
Hmm.
for instance, the lung has to process air and oxygenate the air. But if the lung tissue grows uncontrollably, it fills up all the air pockets. So
Hmm.
it does not function as it's supposed to function. and therefore causes imbalance. You get trouble breathing and stuff like that. And
Okay.
that's the simplistic way of looking at it. But there are
Okay.
other factors that it messes up as well,
Like.
like the immune system, and it kind of increases inflammation in the body, and
Mhm.
therefore again, when we go and talk about metastasis, I'll explain how it can be a bit more damaging to the body and why it's such a big risk.
Okay, so would it be right, Karan, then to say that the reason cancer is so dangerous and difficult to deal with is because it can happen to okay, not any cell, but uh, it can
Most, yeah,
happen to most cells. I mean, that may be a very
most tissues
crude,
of body, yeah.
most tissues of the body, right? Uh, and because it can happen to most tissues of the body, leading to uncontrolled growth, there's no sort of one thing that we're trying to attack or to
Yeah,
fix or to solve, but it can be a bunch of different things at different times.
yeah, and I agree with you. And we term cancer as one disease, which is actually not one disease,
Hmm.
because a breast cancer is very different to, say, a brain tumor is very different to a lung tumor, and and the the mechanisms which govern them are quite different. So if you have a drug for breast cancer, does
Hmm.
not mean you have a drug for lung cancer, and vice versa.
Okay, all right. So, although the root cause, in the sense, for all cancers is uncontrolled growth, the treatments would be different based on what type of cancer it is.
Yeah.
Okay, all right. So, on the screen in front of us, what we've got is so we've got bone, lung, brain, liver, and other. Any before we get into bone specifically and maybe the others as well. Any reason why cancers usually, I guess, are associated with these areas or organ?
No, that's a very good question, and we're trying to figure that one out. In about 1882, there was a guy called Paget
Hmm.
who came up with a hypothesis about a seed in soil, saying the cancer is actually a seed, and when it has a fertile soil, an organ where it can grow
Hmm.
well, it
Hmm.
tries and spreads to those organs. So
Hmm.
these offer very fertile soil for the cancer to grow when it spreads, and therefore these are the main organs which actually it prefers.
All right. And if from our previous conversation, I may have this wrong, but I seem to remember that you said that when cancer reaches the bone, I guess it's the most dangerous. Is that true?
Uh, well, I'm a bit partial to bone, so but yeah, it is incurable. So yeah,
Right.
may not
Okay.
be the yeah. So it's bad as long it is bad wherever it goes, and it's incurable in most cases, anyways. But because most of the cases in breast cancer spread to bone, it
Uh-huh.
makes the largest proportion of patients, and therefore, in terms of just sheer numbers, targeting bone is important.
Okay. Yeah, now I know I'm sort of taking you off from your core research, but I hope that's all right because I just want to set the stage for me and others to also understand this. So uh, I know that there are various different types of cancers, and there probably are different ways to categorize these types as well. I'm not aware of all the specifics, but can you talk a little bit about the different types of cancers and what distinguishes them?
So basically, well, the two main types of categories of cancer. One's a solid tumor, and one's a non-solid tumor, which is your blood cancers like leukemia and myeloma.
Mm-hmm.
Solid tumors are all those tumors which are contained, which are palpable, by which I mean you can feel them, and for solid tumors, surgery is one of the main options of kind of getting rid of the bulk of the tumor. Whereas when you have tumors which are not solid, like leukemia and lymphoma, it is spread throughout the body to start off with, anyways. So the two main kinds of divisions of cancers you have.
And in terms of how difficult they are to deal with, is one more difficult than the other, or
Uh,
how dangerous they are?
I'm not sure. I think it depends more than solid versus non-solid tumors. It's about the the site at which it originates, which actually governs how deadly they are. So a pancreatic tumor, which is a solid tumor, is very the survival is very poor for patients. Once you get diagnosed, the survival is about 6 months. Whereas for myeloma patients, which is one of the kinds of tumor which is non-solid, initially you have quite a long survival rate. So it differs from the kind of tumors which we're talking about.
Okay, so it's important to know where the cancer originates and then where it spread to, I guess.
And it's a proportion
Yeah.
of breast cancer patients
Mhm.
which experience spread to bone, lungs, brain, liver, other tissues.
Uh-huh. Okay, I get it. So more than half of breast cancer cases experience metastasis to the bone.
That's right, yeah.
That's right. Okay, and then I guess sort of an
It's
equal
about,
second to brain and lung.
yeah, about 15 to 20%.
All right. And what would these other comprise?
So others usually lymph nodes. So it's not a particular organ, it's just a lymph node next to the site of the breast tissue or
Mhm.
distant lymph node. Usually,
Okay.
most of them is that. About 9%, 10% of that is that.
Okay.
And yeah, majority of them is lymph node usually.
Okay. All right. So yeah, go ahead and talk to us about the slide, whatever you had in mind about this.
Yeah, so as I was saying, that majority of the breast cancer patients experience, if they're not caught early, metastasis to bone. And when it actually goes to bone, what it does is that it causes these kind of lesions in bone. So if you look at your tissue or the X-ray on the left, that's normal hip joint.
Mm-hmm.
That's the femur. That's the pelvic bone, and that's a nice even gray coloring over it. But
Mm-hmm.
when you have cancer which has spread to bone, you get this discoloration, which suggests that there are holes in your bone which the cancer is making, and which is extremely painful, makes your bone break very easily. So they experience multiple fractures.
Mm-hmm.
So that's the kind of damage it does to bone.
And that's a hip bone.
That's the hip bone, yeah. The cancer will not only spread to the hip bone,
Mm-hmm.
it will spread to the spine, it will spread to the any part of the skull,
Right.
ribs, yeah.
Right. Yeah. Sorry for the ignorant questions, but
No,
I'm just trying to figure it out. So this is just an example. The hip bone is
yeah,
an example, probably an example that illustrates it well.
yeah,
Okay. All right. So, um, okay. Let me just ask you this. So, the image to the right, which shows the metastatic effects, I guess.
yeah,
If you compare that with someone who didn't have cancer, then of course we have the normal on the left.
yeah.
But with advancing age, does that create coloration in any case?
Yes, it does. So you must have heard of a disease called osteoporosis,
Mm-hmm.
which is which is not as well is the so osteoporosis is also the weakening of bone, similar to what you're seeing on the metastatic um,
Right.
uh,
Okay.
X-ray,
Mm-hmm.
but it's not as severe, and the kind of timeline for it is much different. So it's a much slower change,
Mm-hmm. So
and there are very good drugs
Mhm.
for it.
All right. So you could move from
But yes, you would get a hip fracture in case if you have osteoporotic bone when you go older. And when we go forward, I'll tell you why. I can show you why what cells actually mediate these processes. You ask me if when you know people have fractures, hip fractures, is the process similar to having metastasis?
Mhm.
And I said the same cells do make the changes, but the metastasis is much quicker than osteoporosis, and osteoporosis is a better understood uh, phenomenon, and there are drugs already available on the market which prevent or help your bone not to form these lesions.
Okay, all right. So, if you were to put a timeline on that, if possible, how long in a I don't know, you take your pick whether it's an average case or a bad case, but how long would it take to go from, let's say, normal on the left to metastatic on the right?
Uh, you can't put a timeline because again there are a lot of factors which govern it. So in some cases you might have heard of relapsing of cancer. Some cases it happens in the first 2 3 years 4 years. Other cases the cancer cells can be dormant for in bone for up to 20 years, and then
Wow.
so it really is. You can't really put a kind of a particular
Mm.
time scale for all patients because
Okay.
it's yeah. And the other problem with metastatic cancer is that unless it shows symptoms, you would not know if you've got metastatic bone cancer or has it spread to bone, lungs, or liver or whatever. So by the time you actually see symptoms, it's sometimes too late.
Mm-hmm. Okay, so you said you could have dormant cancer for many years.
Yeah.
Now, does that mean that you would not be able to detect it at all if there were no symptoms?
Yes, because there would be a couple of cells lying about in the bone or in the brain or in the liver, and just waiting for the right time to kickstart and again develop a cancer in the organ.
Mm-hmm. So there are no tests that
Not yet, no,
no routine tests that it would show up on.
unfortunately not.
Wow, okay. So what if you went looking for it? Are there tests that would show you if there are
So in our models that we use in labs, yes.
okay.
But in people, how would you do it is a bit difficult. It's like finding one cell or 5 cells in someone's
Yeah.
body. It's going to be very tricky
So how would you do that in your lab model?
because we can stain the cells a particular color, and when we take the bones out, we can find if the cells of that particular color were
Mm-hmm.
there or not, even though we did not have any symptoms.
Okay, fair enough.
Should I move on to trying to describe what the different cell types of the bone are?
Yeah.
And because I think understanding how bone works normally
Mm-hmm.
is important, understanding how it's dysregulated in cancer. So I think the
Sounds
next
good.
slide is about what happens in normal bone for you and me. I'll
Sounds
try
good.
and go through it and ask me anything if you.
Yeah.
Um, there we go. So it looks, so this is bone over time. So on your left, by the way, can you see my cursor?
Yep.
Yeah. So if that's your starting point and that's what happens towards the end, and if that's a time frame, and I think the time frame for this process to happen is about 100 to 120 days usually. So from a quiescent stage
Mm-hmm.
back to a quiescent stage is about 120 days or so. So what happens normally in all our bones is that you have a bone. The yellow color is the bone, and there are cells on the surface of the bone called the bone lining cells, which
Uh-huh.
just cover the surface of bone. When you are doing some exercise or activity, you get small micro cracks in the bone, usually, which is but you can't see in the X-ray. It's very small, and those micro cracks stimulate these cells called osteoclasts,
Mm-hmm.
these big red blobs, which then come to those micro cracks and eat those cracks down. And that we call resorption, which is termed here. So what happens is your bone line cells go away, the osteoclasts come here, they eat the bone away to get rid of the crack, and then these cells die,
The
and different
osteoclasts
cells,
die.
yeah, osteoclasts die, and the macrophages, which then mop up the dead debris of the cells. So that's a small phase. And the new cells called the osteoblasts come into the picture, which then build back this bone back
Uh-huh.
to a new bone.
Mm-hmm.
And then these osteoblasts then convert into bone lining cells, reaching quiescence. So this happens in all our bodies all the time
Okay.
to maintain your bone homeostasis in the norm. So maintain it at the regular level. So for instance, if you're exercising, you'll have more micro cracks. So there'll be more of this activity going on, and you get more new bone, which will be stronger, and vice versa.
And so you're saying the body continuously, I mean, the bone continuously goes through this process
Yeah.
from quiescence to quiescence, and it's about 100-day process. Is that what you said?
Yeah, 120 odd day, 120 days, I think.
Okay, and I imagine different bones would go through this at different points in time, or
No, so it happens throughout your body. There are barring few bones in the body which do not have this.
uh-huh.
One of them is your bone in the ear. Uh, but apart from that, all bones would undergo this process all the time. So what it's thought is that in every 10 years, your whole skeleton is actually new.
Uh-huh. Okay,
So,
that's interesting. All right, I had no idea. Hmm.
so it's not a static tissue. It continuously is dynamic. It continuously kind of metabolizes itself and then makes new bone.
Yeah, so I imagine you'll be getting to that, but the previous time we spoke, you did mention that we usually think of the bone as something very inactive,
Yeah.
but it is active and goes through a lot of activity, which is why it's also susceptible to cancerous tissue or cancers, I
Or the
guess.
cancer can take advantage of these cycles.
Right.
And
Okay.
when you talked about osteoporosis earlier, it is the increased activity of these osteoclasts. So you have more of eating of bone, less of building in
Uh-huh.
older age, and therefore the bones become brittle or more porous.
Okay. So a non-cancer related question.
Yeah.
So usually when does that start happening? When does the osteoclast activity increase?
In normal people or in
You know,
people with
in
osteoporosis?
no, in normal, as I guess, as a regular process of aging.
Yeah. So if you're walking, you put some weight on your skeleton
Right.
that causes micro tears or micro cracks in your bones
Right.
every day. So
Right.
it's happening every day. Some part of the skeleton is the osteoclast is getting activated.
Okay, fair enough. So I guess then I mean in osteoporosis.
So these cells are regulated by a lot of hormones.
Mm-hmm.
So for osteoporosis in particular, in women who are postmenopausal,
Uh-huh.
you have lack of estrogen,
Uh-huh.
and estrogen normally keeps these cells osteoclasts in check. So when you have lack of
Mm-hmm.
estrogen, you have increased number of these cells. They're more active, and therefore postmenopausal women are more likely to have osteoporosis.
Mm-hmm. Okay. And what about in men?
So men do not have a typical time for osteoporosis like women do, but it depends on individual people and their activity levels and all that. So,
Okay. All right.
but generally with older age, again, the cycles are slower,
Mm-hmm.
get slower, so there are more cracks building up and less new bone getting built. So with age, you would anyways
Mm-hmm.
get deterioration of your skeletal integrity.
Right. So that 100-day phase sort of gets stretched out
Yeah, stretched
as.
out, or it's not as frequent. So what I was going to go next was that in cancer, so
Mhm.
in normal people, there's a balance between the building of bone and the resolving of bone or eating of bone. And therefore, your normal bone remains equal. So you don't get a change in your
Mm-hmm.
total bone usually.
Right.
But in cases like osteoporosis or in cancer, that balance shifts to more resorption or
Uh-huh.
more eating via the osteoclast, less building, and therefore you get those holes in the bone that you saw in the previous X-rays
Right.
to cause it to fracture or make it more weak to be so that it fractures easily.
Okay. So, okay, I get the point, but can you go to so the little illustrations that you have on the bottom of the slide? Could you
Yeah,
just talk a little bit about the elements of that illustration?
yeah. It's very simplistic of me saying that this is what happens,
Uh-huh.
but when this happens, there's a lot of regulation to make sure it doesn't go out of hand. And all these boxes, which are shown here, are different molecules or proteins
Mm-hmm.
which regulate the activity of the different cell types. So, for instance, osteoblasts,
Uh-huh.
the bone forming cells, release a factor called rank ligand, which
Uh-huh.
is which actually causes the formation of osteoclasts.
Okay.
So this is to make sure that the activity of both osteoblasts and osteoclasts are coupled together, so
Mm-hmm.
that they remain. So that when you have more osteoblasts, you have more osteoclasts, so that it remains in check.
Okay.
And these are other factors like OPG is an inhibitor of rank ligand, which makes sure that rank ligand remains in the same concentrations usually. Stop me if you're losing me.
No, no, I get
It's
it.
just
So,
all the it's just all the regulation that these cells do towards each other.
yeah,
And it's
so in the larger picture, if we step back, then it's the the quiescence sort of stages that you showed us earlier. But then when you break that down to smaller processes and what's really going on, that's what you're illustrating at the
Yeah,
bottom. Because
yeah,
if, let's say, one stage like osteoclasts, uh, or if the activity of osteoclasts goes out of hand, then that will be bad news. So everything needs
yeah,
an inhibitor and needs to be regulated. Okay, all
yeah.
right.
So, so now we know that how the bone functions usually. I think the next step is to see what the metastasis process is
Mhm.
generally, and then we'll speak a bit about how metastasis occurs in bone. So I
Perfect.
think the next slide is the metastatic process for most solid tumors.
Uh-huh.
So if I can take you through this, if that's your primary tumor, it can be breast cancer or prostate cancer or pancreatic cancer, whatever cancer, and it's
Mhm.
basically a clump of cells
Right.
wherever the tissue is. Uh, it has to it has to invade the surrounding tissue. So that's the first step to break out from the constraints of the surrounding tissue
Mm-hmm.
to reach towards a vessel,
Mm-hmm.
which is this bit here, a blood vessel.
Mm-hmm.
And once it reaches the vessel, it has to pass through the vessel walls, go inside the vessel, travel to a distant organ.
Mm-hmm.
When it reaches a distant organ, come out of the vessel, and then colonize the vessel and start growing there. And that's the whole process of metastasis,
Right. Right. Okay.
right?
Uh-huh. Okay.
So
So,
imagine if it's breast cancer and bone, it has to go from a vessel near the breast tissue via circulation to bone. When it reaches bone, it has to go out from the vessel, come out, find a suitable area, and then start growing there.
mm-hmm. Okay. So this, uh, typically, what's this vessel?
Vessels are any of your blood vessels. So your
All right.
veins or your arteries, or it's mostly veins, but yeah.
Okay. So any blood vessel. Okay.
I was saying that when the cells actually extravasate from the tissue, from the blood vessel, and reach a target organ, especially bone, what happens is that those cancer cells secrete factors which promote the activity of osteoclasts, the bone eating cells.
Mm-hmm.
Now, when the bone is eaten by these cells, it releases, there are growth factors which are embedded in the bone itself, which are released because as the cells eat the bone, it releases factors which then feed the cancer cells to grow. And
Oh,
that's
okay.
that's called a vicious cycle. So the more cancer cells you have, the more osteoclast activity you have, the more growth factors are released, which then again feed the cancer cells and goes on. And that's known as the bone cancer vicious cycle. And it happens not just for breast cancer, it can happen for prostate cancer, which goes to bone, or any other cancer that goes to bone.
Okay. So, when you say the factors are secreted because of the osteoclasts as well, and then it feeds back into the cancer cells,
Yeah.
is this all in, for instance, the bone? Is this all happening within the bone, or is
Yeah,
this
it's
between
happening
the
on the
bone?
bone surface. Yeah.
Right. Okay. All right. So it could,
And
we're talking about bone right now, but it could be non-bone locations as well.
no, because these cells are limited to bone. So,
Hmm.
but some other kind of similar kind of things might happen in lungs if it goes to lungs, but it's a different,
Okay.
these cells don't exist in lungs. So it's a different
Right. Yeah,
mechanism.
yeah, 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 yeah. Okay, I get it. Okay.
And also that the cancer cells suppress the activity of osteoblasts, the bone making cells. So it again tips the balance towards resorption or eating
Hmm.
of the bone, and therefore you get those lesions that you saw in the X-rays.
Right. Okay. Okay. So is this a factor, or is this an aspect of all cancers? This
Most
vicious
cancers that
cycle?
go to bone, yeah,
Okay, so most cancers that go to bone go through this vicious cycle. I guess
yeah,
that's what makes them more dangerous as well.
yeah.
Okay. All right.
So where my work fits in is
Mm-hmm.
that uh, all this was known about that once the cancer cells actually reaches the bone environment, this is
Mm-hmm.
what happens. But
Right.
what my group had found about 5 years ago was that there's a new molecule called LOX,
Mm-hmm.
which is actually secreted by the breast cancer cell when they're in the breast tissue,
Okay.
before they actually leave the breast tissue in the blood vessel. And that molecule travels to the bone tissue and already starts increasing osteoclast activity. So before the cancer cells actually arrive in the bone,
Hmm.
the tumor secretes factors or proteins which reach the target organ, which is bone in this case, and makes the environment suitable. So when the cancer cells do arrive, the growth factors are already there for it to grow.
Wow. So it's like an advance party being sent out for.
Yeah, just to kind
uh,
of seek out, make sure everything is going to be all right when we get there.
yeah, and prepare the scene for the actual danger to arrive. Okay. So this is
So
what you guys found out through your work.
yeah, this was one of our my PI, and so I was part of the group who worked on that to kind of establish that mechanism.
Sorry, what's PI?
PI is uh, PI is principal investigator. Sorry.
Okay. Right. Okay, so you were part of that group which found that out. So LOX is a molecule, you said?
Yeah, LOX is actually a protein,
Okay.
and its basic function in normal physiology is normal in normal circumstances is to make sure that the tissue outside the cells,
Uh-huh.
which is called the extracellular matrix, the proteins and all in there are cross-linked, so there's
Uh-huh.
enough rigidity in
Mm-hmm.
the environment. That's its normal function.
Okay. So when it travels here to the bone and creates the I guess the soil for this the primary cells, tumor cells to come in and do its thing,
Yeah.
is is that function sort of enhanced by its usual physiological function, or does it now do something completely
Uh,
different from what it
no, that's a
usually
good
does?
question. No, it was we found out it does something completely different. So we found out that this molecule enters the osteoclast cells
Uh-huh.
and makes them more active. So it does
Mhm.
something. We're not sure how it does it yet, but it enters the cells and it makes it more active.
Wow, so that's really interesting, right? Because again, it goes back to that soil and seed analogy where it doesn't quite have just one function, but its function
Yeah.
can change depending on where it is. Okay. Okay.
So if I take you back to the previous slide, so you see these green dots here. So
Yeah.
what we feel is that the primary tumor, before it actually goes into circulation, releases these factors, locks in our in
Uh-huh.
what we focus in,
Uh-huh.
which actually goes via circulation to the target organ and prepares the target organ, and that term is called the pre-metastatic niche
Okay.
because the metastasis has not happened yet because the cells have not reached. It's
Right.
something which has happened before the metastasis,
Mm-hmm.
and that's what we're focusing on currently.
Okay. Great. So in the larger scheme of things, what does this finding mean for bone cancer or metastatic research?
So it means that uh, with technology, we have been able to manage to kind of detect circulating tumor cells nowadays. So if you have a blood test, and you in some cancers you can find, if you had cancer you can find the circulating tumor cells,
What
but
are circulating tumor cells?
so once they actually reach the vessel,
Uh-huh.
they're circulating in the body before it actually goes out of the vessel.
Mm-hmm.
So that is called circulating tumor cells because it's going via the blood
Mm-hmm.
to different parts of the body.
Right.
But what we feel you can do is, if you know, if you can understand the mechanisms by what molecules are secreted, you can probably catch tumors early, because if you can detect locks in this in the blood vessel, then
Right.
you know that that might cause that the chance of having bone metastasis is higher. So let's monitor the bone more closely. Let's pick it up earlier. Or
Mm-hmm.
can we do some drugs which target locks, which prevents the formation of the pre-metastatic niche? So those kind
Okay.
of things, and delay metastasis altogether.
Okay, so you're saying we can already, as of today, through certain blood tests, find circulating cancer cells, tumor cells?
Yes, in
Uh,
certain cancers, not all cancers.
okay, in
Uh, the,
certain
the,
cancer.
yeah, and I don't know if it's pathologically as a lab, you can do it, but in labs we can do
Right.
it for sure.
Right. Okay. So now what I guess your research means, or your group's research means, is that you maybe want to see if we can, or this would lead to maybe trying to find out whether you can also find locks in circulation
Yeah,
in an attempt to preempt the spread of the cancer to the bone.
yeah, and
And
catch it
then,
early.
right, to catch it early. And then if that were possible, then you'd be able to maybe talk about drugs to treat it at an early stage before the
Yeah,
complete metastasis has taken place.
yeah,
Okay, and that's very important, I guess, from a medical perspective, because as the cancer moves and metastasizes, I don't know if I got that right. Uh, so as it moves, it becomes more dangerous, especially when it gets to the bone. So if you're able to block that move, I guess that's important medically speaking.
yeah, and it will increase the lifespan of patients who have breast cancer to start off with,
Right.
and because cancer generally, or a human body generally, is quite it 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 does not do different, but it's very conserved mechanisms usually.
Mm-hmm.
So this might be applicable in any other cancer which goes to bone as well, just not breast cancer, because the mechanisms usually remain conserved.
Right. Okay.
So
So.
understanding this process can
Uh-huh.
have bigger applications, just not for breast cancer, for prostate cancer, um, for pancreatic cancer, for other cancers that can go to bone.
All right. So when you say the body usually conserves, you mean that it's usually the same types of processes that take place in different circumstances or for different cancers and activities?
Yeah, because the cells are the same in
Mm.
bone, the mechanisms by which the cancer can control those cells would remain generally the same or in a
Mm-hmm.
similar kind of area.
Right.
That
Okay. All right. So picking up from the research that your group did, has there been further activity on this to maybe
Yeah,
apply this?
so this story about locks has been published now,
Mm-hmm.
but what I'm doing now is trying to understand how locks actually gets from the primary tumor to bone itself. Why does it not go to lung? Why does it not go
Uh-huh.
to liver? And if it does go there, does it do anything there as well? So this image is from an experiment, and
Uh-huh.
that's normal bone. And this is just from some cancer cells. We took some um, media or some kind of supernatant. So when we grow cells, you can collect the liquid that it's grown in.
Uh-huh.
So there's no cells in it, just everything that the cells have secreted.
Right.
And when we add it to our experimental models, you get the same lesions that you would get when the cancer cells are there, showing that before that the cancer cells themselves secrete factors which cause the lesions.
Wait. Can you repeat that?
Yeah. So uh, what we did was we grew cancer cells
Okay.
and we
In your lab, of
collected
course.
everything that the cancer cells secreted or kind of released into the
Uh-huh.
in the environment,
Uh-huh.
and then we just, without the cells being there, we just put
Oh.
what that secreted or released, and what we see is that only what has been released causes this effect in bone,
Uh-huh.
compared to uh, without any cancer cells being there.
Okay, so if you have to simplify this process in terms of like the physical mechanics of how this would be the experiment, so you grow cancer cells
Yeah,
which secretes whatever it secretes,
yeah.
and then how do you expose the bone to that?
So this is done in mice
Okay,
models.
okay,
So we inject the secreted proteins from the cells into mice, and what we observe is that we get these
mm-hmm.
lesions in bone,
All right.
and
So you don't
and
even
to make
have the actual cancer cells,
cancer
but
cells
just
there.
the
Yeah,
secretions are enough to have to create the lesion.
yeah, and to make sure, and this is just to show you that how science is happening. To how do you know that LOX is actually
Right.
doing what it's doing in this
Uh-huh.
case? Is what we did was we targeted. So this graph here shows that if you just have a tumor which secretes LOX, you
Okay.
get increase in these lesions. But when you have an antibody which binds to LOX so that it inhibits the activity of LOX, you get decrease in that lesion number. And furthermore, and that's that bone. And when you stop the cancer cells from actually producing LOX, which you can do that genetically manipulating it, you get even further decrease in your lesion number. So this shows that the effect that you're seeing here in the bone is because of LOX.
Wow, so this is pretty exciting stuff, I imagine, for you.
Yeah, it was good. This is what led to my current research. This is a data that we had published earlier. And,
all right, so you can obviously all this is done in an experimental models now, which means that you can control all of these factors.
Yeah.
So you can not only create cancerous cells, but you can use you can also, I guess, introduce antibodies.
So if you know what antibodies, if you're targeting a particular molecule, then you can use an antibody against that molecule.
Okay, all right, and
if they're available.
Right, and could you repeat the last? So the 41.
This one. Yeah. So that is so the way cells produce proteins is that the cells have DNA, and the DNA gets converted to RNA, mRNA, and the mRNA is then translated to protein. So when we say SH LOX, what it means is that we are targeting the mRNA for LOX in the cancer cells. So the mRNA gets degraded. So the cancer cells cannot produce LOX at the same level.
All right.
So it secretes less LOX in the media or in the outside environment.
Okay, that makes sense. I get that. So is this the work that you're currently doing, or is this what led you to what you're currently
doing? This is what led me to my current work, which is probably the last slide, I think. And this is working diagram that I'm trying to understand. So in here you have the primary tumor in the breast here. And I've told you that the LOX is secreted from there, which then activates the osteoclasts and stuff like that, which then secretes the growth factors from the bone, which acts on the bone forming cells and the cancer cells to grow. What I'm trying to understand is that what causes the secretion of LOX in the first place from the tumor cells? Are there conditions which predispose the secretion of this molecule from the cancer cells? And if you understand this mechanism, then can we target at the root itself to limit the secretion of LOX? Because from the previous graph, we show that if we decrease the secretion of LOX, you don't get as many lesions. So instead of trying to target at the bone environment, can we actually stop the secretion of LOX from the source itself? So that's something what I'm working on. And in the meantime, I'm also trying to understand how, once it's secreted, how can a protein molecule travel via the bloodstream to reach a distant organ? In particular, work on osteoclast. Is there some way that it's addressing LOX to go to bone?
So is there some way what is addressing LOX?
The cancer cells are addressing LOX to go to bone.
So how would you go about figuring that out? Like what sort of I don't know, what sort of a direction or signal or whatever it is that you're looking for?
So the process I'm undertaking is there's something new called extracellular vesicles or exosomes, which have been recently quite popular in the research community. So exosomes are small. Imagine a very small cell. So it's a closed membrane-bound vesicle. With vesicle, I mean just a can't think of a normal word. Just like a bubble.
Okay.
And the bubble might have proteins on the surface of that bubble, which might direct it to go to a particular organ. And inside that bubble might be LOX. So it's like an envelope which kind of addresses locks to go to a particular site, and the stamp or the address is the proteins on the bubble surface.
Oh wow. Okay.
Does that?
Yeah, yeah. So how do we know this?
So what we're doing right now is we are growing these cancer cells, collecting these uh, vesicles from them,
Mm-hmm.
like we did earlier, collecting all the secreted proteins.
Right.
We can collect the vesicles, then we can try and characterize them using very um, um, precise, or not precise, very sensitive techniques of mass spec, which is mass spectroscopy,
Okay.
to see what proteins are there on the surface, what's inside these vesicles, how
Mm-hmm.
it changes with different cancer types, and do these vesicles have a tendency to home to particular organs in the body and stuff like that.
Okay. So how far in are you on this research?
So I've got some preliminary data actually, and I have put that uh, for a grant application. So to get more
Mhm.
funding to take it further. So I have preliminary data which suggests that there are particular kind of vesicles secreted by cancer cells which might mediate these effects. And I have found a subpopulation of those vesicles, and I've applied for more money to see if I can characterize them further and use them to predict the site of metastasis for all cancers, not just breast cancer,
Mhm.
and B, try and target um, the secretion at source to see if I can delay metastasis.
Okay, so can you talk a little bit about these two implications? So you said the first one was try and find out, um,
The predict the site of metastasis.
predict the site of metastasis. Okay.
So predict which organ the metastasis is going to take place. Because interestingly, most cancers initially just metastasize to one organ
Uh-huh.
or maybe two organs. So if it has bone, it's unlikely if it'll go to liver immediate at the same time. So
Right. Okay.
there's an inherent propensity of the cancer cells to go to one organ or the other. So if you understand that mechanism,
Mhm.
you can predict which side it's going to go to. If you predict it, you can monitor it better and get interventions better, quicker into patients. Because right now, most clinical uh, the clinical guidelines is to monitor them yearly until they have symptoms, and they don't really monitor them by imaging
Right.
because you don't expose them to X-rays and stuff like that.
Uh-huh.
So if I can have a blood test which can predict where the cancer is going to spread, and you can take that regularly,
Mm-hmm.
then when something changes, you can look at cellular
Okay.
oncogenomics.
So I think I get that. So currently, how do we test for cancer?
Primary cancer or metastasis?
Metastasis.
By symptoms. So if you present with, say, pneumonia-like symptoms
Yeah.
or breathlessness, you might have a lung tumor, or if you have bone pain,
Yeah.
you would have bone cancer. And generally, the primary tumor is usually identified prior to metastasis.
Uh-huh.
So if you have had breast cancer and you have bone pain, they'll tell you, well, as soon as you have bone pain, just go into the clinic as soon as you get it.
So
Because
how do you test for primary?
primary is so depending on the cancer. So for breast cancer, you have mammograms.
Yeah.
For lung cancer, again, breathlessness symptoms usually.
Okay,
For
but symptoms won't confirm cancer, right?
no, once they have the symptoms, then based on other things, they might do a imaging, like they might do an
Yeah.
X-ray or a MRI to see if there's a tumor. If they do see a tumor, it may not be cancer still. It can be a non-cancerous tumor, which
Uh-huh.
is just a lump of cells growing,
Yeah.
and that's fine. You can just take them out. There's no cancer, but they might need to biopsy it
Mm-hmm.
to make sure it's cancer.
So did you stop sharing?
No, I didn't, but it
Could you
went off.
just reshare if possible?
Yeah.
So what is a biopsy? And the reason that I'm trying to understand this better is because, so you said work of the kind that you're doing could lead to maybe a blood test being used in order to preempt um, cancer, I guess pre-cancerous or pre-metastatic activity.
Yeah.
Um, and so that, of course, clinically gives you a lot of options, right? Because you're preempting it, and you don't
Yeah.
have to wait for symptoms to show up in the bone or wherever it may be.
Yeah.
So, so the reason that and I got the impression that you were saying that most tests right now are imaging tests, whereas we want to explore the blood test route.
Yeah. Because it's easier, less invasive,
Right.
no radiation. Yeah.
Okay. So then, how is a biopsy done, and what is a biopsy?
So biopsy is basically taking a small bit of the tumor tissue from, and biopsy is usually done for the primary tissue,
Mm-hmm.
uh, primary cancer. And what it does, it depends on where the cancer is. You might stick in a long needle to reach it, or if you're taking a surgery to
Mm-hmm.
take the tumor out anyways, that's also a biopsy if you take a bigger of it. And what biopsy does is they you characterize. So once you look at what you can do with the biopsy is that you can see the cells and see if they're cancers or not. So there are
Mm-hmm.
different cancer cells have different markers on them. A cancer cell
Right.
would look very different under the microscope compared to a normal breast tissue.
Right.
And so when you do a biopsy, you can see that difference, and it confirms that you have a cancer.
Okay. So if I get this right, in a cancer diagnosis, let's say you present symptoms that you know the clinician or the specialist or the doctor, whoever, may suspect to be cancerous, and so caused by cancer, and then you get various sorts of tests done uh, for the primary cancerous tissue.
Yeah.
And once that is confirmed, then you have to worry about metastasis. Uh,
Usually, it depends on what stage the
right.
patient comes to the clinic.
Yeah. Yeah. So
But
let's say it's an early stage. So it hasn't
yeah,
there hasn't been metastasis as yet. And so what you'd have is then the second phase of diagnosis, which is the diagnosis of metastasis, for which you're saying you usually have to rely on imaging because that's when it shows that the bone has been affected or degenerated.
yeah,
And symptoms ties in here because you only go for that imaging if you experience symptoms, right? Because
yeah,
you have symptoms and then you figure out, oh, maybe we should test this, and then you get the
yeah.
imaging done.
That's right.
All right. So what you're saying is maybe with this kind of work, we don't have to wait for symptoms to be experienced and therefore start imaging, but we try and do regular tests once we know that there is cancerous activity. So we do
That's true.
regular blood tests which are not invasive, so that gives sort of clinical space and time to for intervention.
Yeah,
Does that make sense?
yeah, exactly.
Okay, now I get
Just
it.
a small correction because I'm a bit pedantic.
Yeah, yeah, no.
Nowadays, we know that it's not as clear cut as you have primary tumor and the timeline for metastasis is different. What recent research
Mm.
has found is that as soon as you have the growth of a primary tumor, metastatic cells are already in circulation.
Uh-huh.
If
Okay.
if it may home to some place or not, it may grow there or not, but there are cells which can be detected in circulation very early on.
Okay. Okay. All right. So we don't, in a sense, have that space or clean cut between
Yeah.
primary diagnosis and then metastasis. Okay. And was there a second thing you said, a second implication of your research, or does that cover it? Does that
And
cover it
no,
all?
the other thing is that, yeah, the second implication was that if I do understand how that bubble or vesicle goes to a particular organ,
Mm-hmm.
I can stop it either going there
Yeah.
or I can stop it from being secreted in the first place from the primary tumor,
Right.
which may then also delay uh, the
Mm-hmm.
metastatic process.
Okay. Okay. All right. No, I get it. This is fascinating stuff, Karan. I've learned learned a lot about cancer in, I think, what's 45 minutes to an hour. So this is this is really great stuff, and I think this is uh, these are terms that everyone hears all the time, cancerous and tumors and biopsies and whatnot. But at least I didn't quite know the specific significance of what it means, and it's great to know what's happening. I guess this is cutting edge research, so it's great to know, uh, find out a little bit about the kinds of things that people like you do, which is extremely important because I imagine so someone like you is working in a lab, and then you have some findings, and then it takes a really long time for that to then find its way into sort of clinical medicine as such. And so we wouldn't probably hear about this for, I don't know, 10,
Yes.
12 years, especially when drugs come into the picture, because I guess you have drug testing and various other processes that it has to go through. Yeah,
Yeah.
so that's great. So I had a question about, because we're in this whole COVID situation and there's a lot of talk about vaccines, and everyone's excited to know more about vaccines. We're not just, we're just hearing more about vaccines in general and not just
Yeah.
about COVID vaccines. Um, so I'm interested to know, in the case of cancer. So I don't know, speak to, I guess, breast cancer, if it makes sense to narrow it
Yeah.
down. Does it make sense to even ask the question of, can there be a vaccine? Or am I just missing something fundamentally different about cancers because of which they can't have vaccines?
And the problem with cancer is different people have different cancers.
Mm-hmm.
So, um, not in terms of different cancers like breast and lung, but even in breast cancer, the phenotype, by which I mean the characteristics of the tumor cell that is actually cancer, is very different
Mm-hmm.
from someone else who has a similar kind of breast cancer.
Right.
And the vaccine depends on identifying the tumor cells as something which is not right, and so that the immune cells can actually um, kind of deal with it.
Mm-hmm.
So, as of yet, we have not found a single molecule which has been common between the different cancer subtypes so that we can use it to prime our immune system using a vaccine. Research is
Right.
ongoing there, and hopefully, when we understand the processes better, the mechanisms better, different subtypes better, I don't think there might be one vaccine for breast cancer, maybe multiple vaccines based on the different subtypes of breast cancer that you have.
Right. So, but the problem that would arise then would be that, because there are so many types of cancer, and you said cancer is not really one disease, right? We spoke
Yeah.
about that earlier. Then the question arises of who gets what vaccine, right? Because then you'll have multiple vaccines for different types of cancer.
Yeah. So, what you think of a vaccine as a preventative thing might not be what
Mm.
we use in cancer.
Mm-hmm.
What we use is something that actually increases the immune response towards cancer
Right.
once you know what cancer it is, so
Mm-hmm.
that the body can fight cancer better as well. Because the body
Right.
does fight cancer. It's not that the immune system usually does identify cancer as something which is not right and tries to control it.
Mm.
But most cancers make sure that they overcome that by secreting, again, the secrete factors which suppress the tumor response, the immune response. So
Right.
if we can identify uh, the molecules which can actually govern the immune response, we can make them more susceptible to the immune activity of our body.
Okay, I get that. So I guess the two different types of ways of thinking about vaccines. One is like a complete preventative measure, and one is sort of to bolster the already existent
Yeah.
immunity that you have against that
And if I'm
body.
not mistaken, I might be mistaken, or I think I've heard this, that what they do is, or what they're trying to do is, they take the cancer cells from your biopsy,
Hmm.
they characterize them because that's personal to you, make identify proteins which are on that, and use that same thing to boost your immune system. So it's each person gets a tailored therapeutic drug, if you
Right. Yeah, I've heard a little bit about this, just here and there, but yeah, I don't know. I don't know anything really significant about that. But yeah, I've heard that. Um, okay, so, but are you, in a sense, as a cancer researcher, are you hopeful of, let's say, a universal vaccine of some sort
for cancer.
for cancer?
No, I don't think that's going to be possible.
That's because we
It's
haven't
too
identified anything.
Yeah, and it's too. The more we know about cancer, it's way too heterogeneous
Hmm.
a thing to have something which is a commonality between them, except that they grow uncontrollably. And the
Right.
reasons for them growing uncontrollably are also different for different cancers. So
Yeah. So I guess in layperson speak, we do refer to all cancers as cancer, but I don't imagine that's how people in the field refer to it. You guys probably refer to each different distinct type of cancer in a different way in any case. So I guess even research then is not going to be focused on cancer because like there's no
yeah,
such category as such. But you'll only have specific work within cancer. Okay. All right. So we've done quite a bit of discussion on your research, and I think it was great to sort of just dive in and hone in on just that aspect of what you do because I learned a lot, and I'm imagining if someone's listening to and watching this, they'd learn quite a bit as well. So I think what we'll do is maybe call it a day for today for this and digest this information, and maybe I can always get back to you and ask some questions that
yeah,
we
sure.
can incorporate at a
Uh,
later point in time.
sure. I hope I was clear enough. I try to make it as clear as I can. I'm not very good with explaining
No, I think it
concepts.
was very clear, and I think these slides really helped because we spoke about getting these slides done. You had suggested it, and I think it really helps sort of clarify the picture because a lot of the terms that you use are things that I have already forgotten, even though we just spoke about it, but I can imagine the diagram. So I think the visuals are very helpful. No, thanks a lot for that. Thanks for
Thank
doing
you
this,
for having
Karan.
me.